The iron jaw. Early jaws displayed deep sockets and wide arches, engineered to handle raw roots, tough fibers, and sinewy meat. Third molars absorbed much of the chewing force, supporting a diet that demanded maximal mechanical processing. As cooking became widespread, food softened, and jawbone architecture responded. Arches narrowed, bone density dropped, and teeth no longer required full functional leverage. Modern jaws often struggle to fit wisdom teeth, revealing a mismatch between inherited structure and diminished mechanical demands. The shift created a paradox. Evolution retained the teeth while reducing the space for them, leaving functionality partly dormant. This biomechanical trade mirrored digestive changes elsewhere, where softer diets reshaped gut length and microbial composition. Fire and tools replaced chewing power with technology, illustrating how cultural innovation redirected physical evolution. And in every modern mouth, the remnants of these forces hint at a past that required more chewing than our bodies now demand. Pain tolerance. Ancient skeletons revealed repeated fractures, deep lacerations, and joint damage, showing survival under conditions without medical care. Genetic markers controlling endogenous opioids appeared highly active, suggesting stronger internal pain regulation than most modern humans possess. As communities developed safer environments, predictable food, and medical treatment, the selective pressure for extreme pain tolerance relaxed. The body's threshold lowered while cultural systems assumed the role of injury mitigation and protection. This shift exposed a trade-off. Genes remained, but expression weakened, leaving latent capacity in place without functional necessity. It mirrored other environmental buffering, like reduced cold exposure softening brown fat selection. And current research probes whether these dormant pathways can be nudged, hinting that natural tolerance is partially preserved beneath modern comfort. The climber's foot. Early foot bones revealed a flexible midfoot, robust toe tendons, and a big toe angled inward, capable of gripping branches and uneven surfaces. Those structures supported climbing, rapid vertical escapes, and precise balance on complex terrain. As bipedal locomotion became dominant, arches formed, toes aligned, and flexibility diminished, trading climbing ability for endurance and stability on open ground. The change shifted energy allocation. Muscles once dedicated to grasping now supported sustained, walking over long distances. The foot's new design reinforced a broader evolutionary pattern, where mechanical structures adapted to the most frequent demands of daily survival. This transformation mirrored changes in other systems, like sweat-based cooling and jaw reduction, showing how efficiency reshapes anatomy. And beneath every modern step, remnants of the old grip linger silently, unused yet still embedded in skeletal structure. Raw fiber digestion. Early human guts were long and flexible, supporting a diet of raw roots, dense fibers, and uncooked vegetation. Archaeological residues on teeth and stone tools confirmed frequent reliance on abrasive, slow-digesting plants. As cooking softened food, gut length gradually decreased, reducing the need for extensive fermentation. Microbial communities shifted toward diets that required less mechanical and chemical processing. Energy once devoted to breaking down tough plant matter was freed, allowing faster digestion but a diminished capacity for raw fibers. This mirrored jaw and tooth reductions, showing coordinated evolution between mechanical and digestive systems. And current microbiome studies suggest that ancient fiber processing capacity may be partially recoverable through dietary interventions and microbial manipulation. Body hair. Skin impressions from early humans showed follicles spaced far closer together, forming a coat that buffered sunlight, heat loss, and constant insect exposure. As climates shifted and movement patterns changed, another system grew more advantageous. Dense sweat glands that cleared heat faster than any layer of hair could. Follicles reduced in size while sweat capacity expanded, creating a body optimized for long-distance travel under high temperatures. This produced a trade in biological priorities. The cooling advantage outweighed the insulation the old coat once provided. Fire, shelters, and clothing reinforced that shift, taking over the tasks that hair had managed without assistance. The thinning coat also fit a wider pattern in human evolution, where energy moved into systems that supported endurance rather than maintaining older thermal defenses. That same redistribution helped shape later cold adaptation changes, showing how climate pushed biology in multiple directions across different eras. And the scattered follicles that remain today work as small signals of a cooling strategy that replaced insulation rather than building on it. Magnetoreception. Human cryptochrome proteins found in the eye responded to magnetic fields under controlled experimental conditions. Some animals use these proteins for navigation, raising the possibility that early humans relied on a similar system. Ancient migration patterns suggest selection pressures once favored this subtle sensitivity to Earth's magnetic field. As tools, landmarks, and social mapping replaced innate navigation, the biological trait faded in prominence. Molecular traces persisted even though behavioral evidence remained weak, leaving a dormant system encoded but rarely expressed. The pattern mirrors sensory reductions in smell and directional hearing, showing that decline doesn't mean disappearance. And current research continues testing cryptochrome responsiveness, probing whether latent magnetosensitivity still shapes human behavior. Cold resistance. Skeletal remains from cold-adapted humans revealed compact builds, wide nasal passages, and rib cages optimized for dense lung capacity. Preserved tissues contained unusually thick layers of brown fat, specialized cells that converted energy directly into heat. Over generations, fire, clothing, and insulated shelters reduced exposure to freezing temperatures, removing the pressure that maintained these adaptations. The biological defenses weakened, leaving most adults with only a modest thermogenic response. This shift paralleled the loss of dense body hair, showing how climate-driven pressures gave way to cultural solutions. Energy once dedicated to thermal regulation was redirected to endurance, mobility, and brain maintenance. And ongoing studies now test whether dormant brown fat cells can be reactivated, hinting at a latent capacity still encoded in the body. Third eyelid. A small ridge inside the eye socket had once anchored a moving membrane, and its position revealed a function most modern eyes lost without erasing the trace. That ridge matched the path of a translucent film that slid over the cornea during sudden wind, dust, or impact, giving early hunters a protective layer that worked without interrupting sight. The membrane's architecture remained detectable, yet its muscular control had weakened as genetic switches for movement fell into disuse. The structure persisted as a silent fold because the body never removes unused parts quickly. It lets them drift until selection stops reinforcing precision. This drift created a contradiction. The membrane stayed visible in anatomy but no longer performed the role that once justified its complexity. The loss didn't signal decline. It signaled that external protections, shelter, crafted coverings, and more predictable environments took over the workload. The same pattern reappeared across other senses, where genes stayed present but slipped into inactivity once better substitutes emerged. And this quiet fold at the eye's corner became the first clue that human evolution favored energy reallocation over maintaining specialist defenses. Its presence opened a wider question about how many sensory tools still exist in partial form, not gone, just waiting for the pressures that once kept them active. Superior nose. Genetic scans revealed an unusual pattern. Long stretches of olfactory receptors remained in the genome, yet many carried mutations that signaled decades of inactivity. Ancient DNA showed these same receptors under strong selection, suggesting a period when subtle chemical cues shaped movement, hunting, and food choice with far more precision than they do today. As tools for distance tracking improved and visual cues took priority, the pressure to maintain vast scent-based mapping declined. The receptors stayed in place but accumulated errors, turning functional genes into fragments that recorded a role no longer reinforced. This created a split identity in the sense itself. A once dominant system survived only as scattered remnants while vision grew into the main navigational guide. The transition also exposed a deeper trend, where cultural strategies replaced biological acuity and shifted the sensory hierarchy rather than expanding it. This narrowing wasn't unique to smell. It paralleled other declining sensory channels that once carried survival importance but later became secondary. And the dormant receptors raised a question that extended beyond scent. How many other navigation tools dissolved once humans relied on external references instead of internal perception? Ear mobility. Early skulls revealed strong muscle attachments around the outer ear, engineered to pivot the pinnae for precise directional hearing. Those muscles allowed humans to detect the origin and distance of sound without moving the head. Over generations, reliance on head rotation and cognitive processing reduced the functional need for these muscles. The attachments persisted, but neural pathways controlling them weakened, leaving only vestigial remnants. This decline mirrored other sensory losses, like smell and magnetoreception, showing a consistent pattern of reduced biological priority. Energy and control shifted to cognitive interpretation, not mechanical movement. And the dormant structures hint at reflexes that exist but are largely unexpressed, raising questions about latent capacities still encoded in our anatomy. So, what are researchers doing now? Laboratories tracked brown fat activation in adults exposed to controlled cold, testing whether dormant thermogenic systems could respond. Neuroscientists applied non-invasive stimulation to explore modulation of pain perception along pathways once critical for survival. Microbiologists engineered gut communities to enhance fiber breakdown, probing whether ancient digestive capacities could be partially restored. Biophysics groups measured cryptochrome reactions under shifting magnetic fields, seeking evidence of latent navigation sensitivity. None of these studies aimed to fully resurrect lost traits. They mapped how modern physiology still reacts to signals shaped by ancient pressures. The work revealed a pattern. Evolutionary reductions are not erasures, but shifts in responsiveness and allocation. And across all systems, thermal, sensory, digestive, and neurological, traces persist, partially active, quietly encoding the history of human survival strategies.